We either add pollen that has an alkaloid in it, a plant-derived alkaloid, or pollen that doesn't have a plant-derived alkaloid on the top there. And then we added some sugar to those flowers. So some of those flowers had defended pollen and some sugar. Some of them had not defended pollen and some sugar. And what you can see over the course of the bout, you can see that bees are shifting their preference. They start out when they come into this naive in a chamber, evenly selecting both plants. They don't have any preference for defended pollen or not defended pollen. But even within 20 landings, so very quickly, this is the first 20 samplings, they're almost 3/4 of the time going to the plant without chemical defenses. And what's key here, and I'm not going to show you all these data and walk you through this whole plot, but like this is very finely tuned. So if we mess with the concentration of sugar or we mess with the concentration of defenses, it has a really major impact on sort of the rate at which bees shift their preference and the strength with which they discriminate. There's some stats if you care. Okay, so we say that they assess to decide what to eat based on rewards, but these rewards are really complex. It's not just like how much sugar is in a flower, it's not just how much protein is in a flower, but there's chemical complexity there. And what I'd argue is there's also all sorts of other complexity that's also hidden, which is microbes in this case. So we saw the sort of visual functional traits. I told you about some hidden chemical functional traits. But there's also microbes living in and on these flowers, and they are really critical both to the traits of advertisement and of reward that plants produce. So I'm going to give you a couple case studies that some folks in the room will be really sort of familiar with because they're their papers. But the first thing I want you to know is that microbes change floral signals. So these are data about a couple common nectar microbes collected by Rob Schaefer, and Rachel was the anchor author on this. And he let some bumblebees into a tube where he was pumping air over one of two microbes in a Y maze, and then characterized how long bees spent in a Y maze for each of these microbes, and showed that initially bumblebees had a really stronger preference for one of these microbes than the other, right? A little more than chance they spent going towards this microbe, much less than chance they spent going towards that microbe. He then let them consume nectar that was conditioned by these microbes, so they did some metabolism in that nectar, and he showed that even though bees prefer the smell of that nectar, that microbe in red, they actually consume less of the nectar when it's been conditioned by that microbe, right? So here is a signal change, here is a reward quality change, right? Bees are showing us they don't like this as much as they like this. And after they've been exposed, when they're no longer naive, we see a clear shift in preference away from spending about half the time in the Y maze for that initially preferred microbe to less time. So this is a clear shift in floral signals, also a clear shift in floral rewards. They also change rewards in crazy ways. So these are data from Rachel also showing that not only do they like grow pretty well in the presence of some of those toxic metabolites, but they also get rid of them. So there's a bunch of microbes here in different colors. This is the concentration of nicotine in a nectar solution with those microbes grown in it. And you can see a couple of these microbes significantly decrease the concentration of nicotine. And one of those microbes, actually, this is the change in microbial abundance growing in that nicotine treatment, does pretty well. So it seems like not only is it getting rid of the nicotine, but it's using it. More data from Rachel shows that there's all kinds of differences in how much nectar bees consume when we treat it with these microbes. Okay, so the microbes, they're making smells, they're changing the way rewards taste. And some data that I collected here with Rachel and with a really awesome undergraduate, Katie Lynch, shows that these changes cascade to impact plant fitness in the way that I told you, right? So we know that most plants are dependent on pollinators. And what Katie wanted to do was inoculate a bunch of different bacteria into Epilobium canum at the, what is it called? The Water Smart Garden out there by the Ag College. And she inoculated these nectar microbes, let the flowers sit open to pollinators for a couple days, and then went back and looked at seed set and seed viability and seed weight. And it was very clear that some of these bacteria are strongly increasing seed pod weight, and in some cases, the number of seeds made. And she did some extra experiments where she actually added pollen onto stigmas. So said, hey, what happens if pollinator visitation went up and there was more pollen moved? And the magnitude of change when she added pollen is about the same as the magnitude of change when we add the microbes. So it seems like the microbes are impacting pollinator preference. So I showed you that sort of, simple bipartite interaction where we have plants producing signals that are advertising rewards. We have pollinators learning to associate these signals with complex rewards. But on top of that, there are microbes who are producing signals of their own, who are changing rewards in the ways that pollinators care about. So that's sort of the framework I'm going to tell you about today. Cool. Okay, so hopefully I've convinced you floral microbes are important because they change plant fitness. And if you're me, and I hope you guys think like me, because everyone thinks like me, right? You might be wondering what processes then shape this microbiome assembly. If it's so important for plants, what controls what microbes are in which flowers, and how predictable is it? Can we go out on the landscape and guess which microbes are going to be where and how that's going to impact fitness? And floral microbiome assembly follows all the same rules of other metacommunities, right? We have these patches of flowers, and then microbes that live in nectar, they're primarily yeasts and bacteria, and they grow best in yeast and bacteria. They can grow in other places, but they do really well in nectar. It's a stressful place to live. It's sort of osmotically stressful. There's not a lot of nitrogen, lots of sugar. And so these act just like sort of textbook metacommunities. We can imagine a community then where a bee comes in and she sees a flower. She senses those signals I told you about and decides whether to land or not. If she lands and drinks, she'll deposit some microbes. We call this dispersal, right? This is like fundamental metacommunity theory. And then as she moves from flower to flower, these microbes disperse, maybe at different amounts. So you can see the green microbe there maybe has a really small dispersal kernel. It's hyperabundant in the first flower, less abundant in the second, gone by the third, whereas maybe the yellow microbe has a longer dispersal kernel. And you would think, like if we cared about this, these dispersal kernels would be really well established, but they aren't. We don't know what these dispersal kernels look like. Like how many landings does that green microbe carry over? How many landings does that yellow microbe carry over? If we really want to try to predict metacommunity assembly, this is one of the foundational pieces of data we need. And this is probably really important sort of at like local scales, but also at landscape scales. But it's pretty hard to characterize for a lot of reasons. I didn't change this after our talk, actually. We were just talking about this. But it's pretty difficult to characterize, mostly because bees are messy and they're would be like feed a bee a microbe and then follow her and see how many microbes disperse to each flower. But her mouth is covered in microbes all the time, right? And so she's not sterile, she's not aseptic. And a bee that is aseptic is not going to act like a regular bee. She needs to be naive, right? And so these are really hard to characterize. And I think the importance is compounded by other metacommunity processes. So Tad Fakami at Stanford has shown that priority effects, for example, are really important in these metacommunities. So who gets to a flower first really really predicts who gets to win in that competition. If you're first, you tend to grow really well, and in a lot of cases have a competitive advantage to any other microbes that come after you. And so I hope that you think that these dispersal kernels are important. That's one of the metacommunity processes that could shape assembly. Additionally, I'm in Zoom. There's post-dispersal processes that could happen. That's where I need to click. And so these pollinators are the main dispersers. When flowers open, they're not sterile, but the nectar microbes that grow in them are really low abundance, probably close to zero. So a flower opens here. Imagine an epilopium flower coming. A bee comes, she drinks this nectar and deposits some of those microbes. And the second process you probably learned about in your like intro to metacommunity theory was host selection, or what do they call it in metacommunities, like site specificity, site selection. So you can imagine that the other thing that could shape which microbes are where is that different microbes grow better in other flowers, right? I showed you that with the nicotine from Rachel's work. And you can imagine that this might be different in different communities. And so this host selection, also called environmental filtering, could be driving this metacommunity assembly. There's also all the things that happen in metacommunities, right? Like within community interaction. So maybe the green microbe is a better competitor than the yellow one. And so its relative abundance grows. And then a lot of my dissertation committee would be mad if I didn't mention stochasticity, which is probably also happening, right? Although some of us are determinists to the core. Okay, so that's how communities assemble. I'm going to show you some case studies now that look at each of those processes. The first thing I'm going to tell you about is asking a project where we ask the relative importance of dispersal versus sort of environmental filtering effects in flowers. That was the one I did in Rachel's lab. Then I'm going to tell you about a project looking at the sort of traits that might drive this host filtering, this environmental filtering. And these are new data from Florida. And then I wanted to really tease apart and figure out a way to phenotype microbes and their dispersal ability. So I'm going to tell you about some preliminary work we're doing there, figuring out if we can actually characterize these dispersal kernels. Okay, so what is the relative importance of dispersal versus host filtering? This is largely a plant and microbe problem. This sort of had two sub-questions. This, like I said, I did with Rachel and with Tobias Mueller, who's now, he's still at Cornell, working on all kinds of bee microbiome questions. And we first asked that first question I told you, sort of dispersal versus filtering. And then, like I said, I'm a determinist to the core. I wanted to know if dispersal, which often in meta-community theory is treated as this sort of stochastic process where the only predictable thing is distance from source population, are there other deterministic factors that shape dispersal, right? Is it more than just how far you are from a source, but are there other things about the sink population that might predict the probability of dispersal? And I was so lucky, like career-making lucky, that when I came to Davis, Rachel had already set up this common garden. And in the common garden out at the Western Center for Water Use, it's really cool. There's a bunch of epilobium. And Rachel did this wonderful study where we have a bunch of plots where the water wasn't manipulated when I was there, but someday will be manipulated. And then we have epilobium canem, which y'all are familiar with probably, right? This is a super common horticultural plant in California. And it occurs from the coast all the way up to almost the top of the Sierras. And so there's tons of ecological variation in functional traits. And that ecological variation in functional traits has been selected by horticulturalists. So we end up with these varieties that are very distinct in terms of their functional traits. And so like lucky postdoc me walked in, we have this big common garden, same pollinators, same soil, same water, and then a bunch of functional traits that are different among plants. And so you can imagine how we might test for host filtering there. What we did is we went out and we bagged flowers. And when we bagged those flowers and let them open inside the bags, there's not very many nectar microbes in them. And then into some of those flowers, we inoculated a synthetic community. If you buy that 2 species of community, synthetic community of 2 species, 1 yeast, 1 bacteria, and then some sugar water, which is what the microbes were suspended in. We did this for a bunch of flowers. And then we rebagged them and we let them grow. And so if we were seeing sort of host filtering, environmental filtering, if these genotypes or cultivars differed, then we would expect that after we let these microbes grow for a couple of days, we would see different densities of our 2 species here, right? And so what I'm going to show you are our 4 genotypes. These are the 4 genotypes or cultivars, horticultural cultivars. And then on the y-axis is the density of yeasts on the left and the density of bacteria on the right. And what we saw was a slight signal of host filtering, right? So on the left, you can see that there's no significant difference in yeast density after 2 days of growth, which means there's no significant host filtering there, at least at the cultivar level. But in bacteria, there was sort of clear host filtering, right? Silver was a pretty poor host for bacteria. Calistoga was a pretty good host. One cool thing here is that each of these points represent the modeled mean and variation around individual plants. So within silver, some plants are particularly bad hosts. There's certainly within cultivar variation, but at the cultivar level, we only saw host selection in yeasts. And we wanted to tease apart this question about which traits are shaping it when I did this experiment, but I didn't do a great job at the HPLC. We did measure some sugar, but sugar concentrations in the nectar weren't correlated with that host selection. So you can see clear variation in sugar concentration, but it didn't predict the final densities of yeast or bacteria. So there's something else besides nectar sugars. Okay, so you told us you're going to tease apart host selection and dispersal. Well, that's host selection. How did you do dispersal? So the way we measured dispersal, or the relative importance of dispersal, is we then extracted nectar from openly visited flowers in the fields. And by comparing those openly visited flowers where natural dispersal is happening to the flowers where we controlled dispersal, right, where we were the ones who moved the microbes around, we can say, are they the same or different in those 2 cases? And if they're different, then dispersal is doing something important in the natural populations. So we did this for a bunch of these plants. And we found that in openly visited flowers, there were big differences in the proportion of flowers that contained microbes. This is the first indication that there's differences in dispersal. So some hosts, had 60% of the flowers with microbes, while others had 20. And that was different for both yeasts and bacteria. But what was interesting is that after we take out all the zeros there, right, these proportions, we do a hurdle model. In the flowers that did have microbes, there were no differences in the densities of yeasts and bacteria. Right? And so it seems like that presence-absence thing is driving the signal in real flowers that are open to pollinators, whereas in our controlled dispersal, where we are the dispersers that are neutral dispersers, there's clear host selection. When we plot the good hosts for yeasts in the controlled experiment versus the good hosts for yeasts in the openly visited flowers, if we were expecting that host selection to be driving a major pattern in the real flowers, we'd expect that good yeast in experiment 1 would be good for yeast in experiment 2, right? Does that make sense to folks? We didn't see that. This took me, You could drop the outlier, it doesn't change it. You can tell yourself it would change it, and you can draw a line, but it doesn't, right? So there's no, but there's no pattern there, right? So this means that when you were a good host for Mechnicovia in the controlled experimental dispersal thing, where we're doing the dispersal, you aren't necessarily the best host for yeast when bees are actually moving around. And so that lets me know that maybe dispersal is playing an important role there, and that it might be even overwhelming the sort of effect of host selection. So I think even though we found clear host selection there, they were rivaled by dispersal, and that perhaps floral traits that shape whether or not a bee lands on you, or a hummingbird in this case, may be more important. So if you're a really attractive flower, then you're sort of shaping your own dispersal, right? Dispersal wasn't random. It was deterministic. Some cultivars had more microbes than others, and that wasn't because they were better hosts, it was just because microbes were getting to them. So maybe dispersal isn't just a stochastic process with distance as the only predictable variable, but rather something about the plants is controlling whether or not pollinators come to them. That's not surprising, and that's a major force in shaping meta-community assembly. Okay, question 1, done. What's the relative importance? I think it's mostly dispersal, but in some cases it can overwhelm host filtering. Now new data, okay? What types of floral traits shape host filtering? So this is work that was done by Jessica Williams and Tony Jordan Millett and Dan in my lab, a postdoc, grad student, and technician out at Archbold Biological Research Station. If you haven't gotten to visit it, it is spectacular. It is a wonderful place. It's not the Sierras, but it is a mountain. It's 100 feet above sea level. Mr. Florida is a mountain. And Aaron David out there helped us with this project. And when Jessica approached me, she was really interested in sort of floral temperature relations. And I had this idea thinking about light transmission in flowers for a long time that I'll tell you about in a second. But light transmission is really understudied. So everybody, when we go out and measure the color of flowers, we shoot a light at it, we let it bounce back into a speck, and we see what light bounces back, and that tells us what color it is. But not all of the light bounces back. In fact, in some species, 70% of the light passes through petals, right? So transmission is abundant and common in plants. Some plants, it rivals reflectance. In other plants, it really outweighs reflectance. And I'm willing to bet that this varies within and among species like all of the other floral color traits we've seen. And I think it's probably particularly important in tubular flowers. And I think this is an ideal sort of candidate for thinking about what floral traits might shape microbiome assembly. And so Jess came to me, and I had remembered a close friend of mine, Chris Moore, who's faculty at CSU Northridge right now. When I went to interview at UNR, Annie and Chris drove me up to Donner Summit, and he was like painting nail polish on manzanitas because he had seen these clear windows on manzanita up at the top. They're not super clear here, but if you look at arbutus, you can see that some members of Ericaceae, this is the blueberry family, have these like totally transparent windows on their urceolate urn-shaped flowers. And he was like, what are they doing? What are they doing? He had an idea about like pollinator-mediated stuff. I had just come from a job where I made maple syrup. And it turns out that when you make maple syrup, you let light pass through the tube to kill microbes growing in the 2% sugar that comes from trees. And I was like, maybe it sterilized them. And I've been holding on to this idea. I pitched it to Rachel. She was like, sure, dude. And then, Florida gave me a couple $100,000. I was like, I'm doing that. And then she was awesome. She wanted to test it in this system at Archbold. So these are two co-occurring ericaceous plants, two Lyonia species. They're related to manzanitas. And she had this hypothesis, which was great, that maybe they're transmitting light and that warms up the flowers and that differentially impacts all sorts of things about them. And I had my pet hypothesis about the UV filtering. So she went out and she shot some light at the petals, measured how much came through in the windows and in the petals, and showed that there's clear differences in UV transmission between the windows and petals. So, this text is supposed to come up. Windows and petals. So the petal portion, the colored portion, lets through very little UV. The window lets through much more UV in both of these species, a little bit more in the white one. There's no difference between the windows and petals in IR, but they're letting through a lot of IR light. So they're transmitting a ton of light, and Fruticosa is transmitting a lot more UV. That's kind of cool. That's like, maybe this is a cool system to test my idea. Then what she went and did is went and put thermocouples in all of the flowers and then right next to them outdoors. So she measured the external temperature and the internal temperature of those flowers simultaneously for 9 plants across 7 flowers per plant for each species with 126 samples. And she found something really cool. This is not a thing I had ever thought about. but there's very clear differences in thermoregulation. So the dotted line there is the 1-to-1 temperature, meaning the flowers are the same temperature as the outside. And you can see that Lyonia lucida, the pink one, statistically indistinguishable from a 1 slope, right? It's a little warmer than ambient, but it seems to be just tracking ambient. Whereas its congener in the same habitats is clearly doing thermoregulation, right? It's warmer than ambient when it's cool out and cooler than ambient when it's warm out for Florida, right? This is cool. And you can see the slope here. So the points are slopes for individual plants again, because we have replicates per plant. And you can see that some plants, again, there's lots of variation. So some plants for lucida, the pink one, actually seem like they are also thermoregulating. So it seems like there's some within species variation, but at the sort of species level, one of them thermoregulates and one of them doesn't. That's what that means. Sadly, it doesn't seem to correlate at the floral level. So if we take all the transmissions for all of those flowers and then map the difference between external and internal, there's a weak correlation there, but we have a lot of samples. We have a lot of power. And so if there is an effect there, it's, weak, if anything. But what is cool is that warmer flowers tended to have many fewer microbes. And this is a strong effect. This is quite a strong effect. And there was a temperature by microbe by species interaction. So temperature is acting differently on the 3 classes of microbes we're studying here, yeasts, molds, and bacteria, differently in different species. So broadly, yeasts are just less abundant in warm flowers. Molds, there's no difference in the slopes here, just in their intercepts. So there's a little bit more mold in lucida, but the slopes aren't different. That's what that arrow means. And then in bacteria, there's a very clear species by temperature effect, where in the white flowers, there's this strong negative effect of temperature, and it's slightly less strong in lucida. So that's kind of cool. She was right, I was wrong. Oh, I didn't tell you I was wrong yet. I was wrong. I'll show you in a second. Only IR transmission was associated with microbial community. So here, dotted lines are non-significant, solid lines are significant. On the x-axis is IR transmission. On the y-axis is on the x-axis up top is UV transmission. And then this is culturable microbes, either by IR or by UV. And what you'll see is there's lots of non-significant effects, except one clear effect where flowers that let through more IR light have many fewer mold spores in them, which is kind of cool. Florida's a moldy, wet place, and this seems like a really neat finding. I'm really excited that she found it. I had this idea, we designed the experiment before we found the results, where perhaps if UV filtering was working, we could take the communities from flowers where I thought UV filtering was happening and then blast them with UV light. And if they'd already been filtered by UV, then the drop should be smaller. It didn't happen. So the plant that lets more UV through does show stronger filtering by UV. So this is presence of microbes in solution before and after we blast it with UV. This is density of the non-zero values. And you'll see that the one that doesn't let very much UV light through shows no change in presence of microbes, whereas the one that lets much more UV light through actually shows a strong drop in the presence of, this is yeasts, molds, and bacteria again. So it shows a strong drop in yeasts and bacteria, which is kind of interesting. So this finding, I think, shows that flowers are microhabitats, and they have this sort of variable internal abiotic conditions that are shaped by plant traits. And in this specific case, floral windows modify light and thermal dynamics. And it seems like assembly is largely driven by temperature, which was exciting. It's like really fun for a student to come, and for her, this was her first project, and it like worked. I was like, this isn't gonna happen every time. This is gonna get harder, I promise you. But like knowing Jess, it might happen next time. She's great. They probably also impact plant fitness, right? So I'm telling you all this stuff about microbes, but like how warm your embryos are is really important. And so I think this could be really important in colder climates. So I'm trying to find some money to send Jess out to the Sierras to work with Chris, because we have manzanita coming from the coast all the way up to the top of the Sierras. There's variation in whether they have windows or not. And I think Chris is really interested in pollen germination rates and temperature. And I think we have a pretty clear effect of these windows shaping internal temperature. What types of traits shape host filtering? Well, one of them is light transmission. There's a lot of other ones too. Okay, last story. And I'm kind of on time, right? Are we doing okay on time? Sweet. So these are the last new data. We wanted to ask about whether microbes sort of differ in their dispersal ability and at what taxonomic scale this variation is partitioned, and whether pollinators might disperse microbes differently, right? And so I hope you bought my case that we should be characterizing these dispersal kernels. This is all work that's happened at UNR or at FAU. I really wish I still lived in the mountains. At FAU, with the help of two awesome undergraduates, Katrina and Fiona, along with Tony, who figured out the method, and Jess, who's been doing a lot of the data collection. And so I was trying to come up with a slide to sort of frame this project before I just jumped into the data. And I wanted to just bounce back to sort of meta-community theory. This isn't the best plot of it, but broadly, meta-community theory tries to classify these cases of sort of coexistence and what's largely shaping the coexistence of species among a bunch of meta-communities. And they try to plot their system into this like dual axis of dispersal and heterogeneity, and then they throw in null models for cranky people. And this idea that like in some cases, we're gonna see strong species sorting. This is the host filtering thing, right? Where like only some species establish in some patches, only some microbes establish in some flowers. But you know, patch dynamics, this thing that takes into account dispersal rates and the likelihood of dispersal among flowers, and I showed you that patches might vary in their likelihood of deterministic dispersal, takes up a lot of this space, this sort of trait space between heterogeneity and dispersal. And so I think really characterizing these dispersal traits is going to be important for predicting meta-community assembly. And so we developed a sort of new pollinator-independent dispersal assay. So what we do is we take microbes and we make cell suspensions of them at all the same density. So you might imagine 2 microbes isolated from a flower, and then we take some sterile nectar solution, and we, oh, I skipped the picture. Take a bee tongue that they're honeybees, they're non-native. We catch them and we cut their tongues off, and then we sterilize them. And then we take that solution and we plate it out, and we can count how many microbes immediately after we dip the bee tongue. We take this solution and we plate it out and see how many cells were moved. And then what's really neat is we figured out a way to sterilize these tongues with a little relay and UV LEDs, and so you can stick the tongues into this chamber and sterilize it. So then we can ask questions about whether tongues differ. So this is a trait about the bees. How different do bees disperse? And we can use that in our models, either as a mixed effect to account for sort of differences in individual, or to test hypotheses about whether individuals vary. And if you were writing a grant, you might say, buy me a robot pipetter and I'll glue them to the pipette tips, and it's all built to fit in a robot pipetter. So someday, if they give me money, I'm gonna do that. And we found, let's just cut to the chase, that there's clear among and within genus differences in dispersal ability. This is Hamelia patens. It looks a lot like Epilobium canum. It's like Florida's version of Epilobium canum. It's horticultural. There's lots of phenotypic variation. It flowers all the time. There's a ton of nectar. So we went out my first semester there and just isolated a ton of microbes. And then did Sanger sequencing to identify them. And for this experiment, we have 26 strains from 10 genera of pretty common nectar microbes. And then we did this dispersal assay, and it was cool. There's like actual differences. So this is the number of CFUs dispersed by different genera. And then each of the points is a replicate for a strain. I think we have like 5 or 6 replicates per strain. But the clear effect here is that there's big variation among genera, but also within genera, strains differ from each other, right? So it seems like there's all kinds of variation that could be exploited in building sort of dispersal models of these microbes. The one bee trait thing, I promised you bee traits, strong tongue effect. Some tongues move a lot of microbes, some don't. And these are all the same species of honeybee. And we haven't like, you're probably like, what is it? It's probably like the number of hairs or the length or how good somebody cut it off a bee, who knows? But there's something there, which is kind of cool. We can group these into kingdoms. So some of them are yeast and some of them are bacteria. And there's a clear kingdom effect opposite of what we find in California, opposite of what the evidence suggests in California, where fungi disperse much better than microbes by like an order of magnitude, right? And this is intriguing because out at Stebbins Cold Canyon, there's evidence that bacteria are much less dispersal limited. They're more abundant, they're at higher densities. Their like probability of not being in a flower is much lower than fungi. It's been a while since I thought about this data. That's the right? Yeah. But I think this could suggest that propagule pressure is really important, right? Because the thing I told you was that we standardize cell densities, and bacteria grow to much higher cell densities than fungi in natural populations, often 2 or 3 or 4 orders of magnitude more than fungi. And so, you know, we got to figure that out. I think this is a new assay, but we're figuring it out. And what I think this probably means is that dispersal probability at the microbes perspective interacts with host selection differently for different microbes. Here, yeasts are dispersing more easily, but this could be overwhelmed by propagule pressure in nature. And, you know, I can wave my hand and say it, context dependence. It probably depends on context, right? What axes of context matter, I think, is an intriguing question. But I do think that, like, I do some models. I didn't talk about any models, but models really need to account for this variation in dispersal. The one last thing I'm going to show you, just because the plots are so cool. So these data are all preliminary data for a grant I just submitted, using some data collected by Mark Dayrup out at Archbold. And he's been there for Archbold. There's no I. Archbold. If you go there, don't say Archbold. He's been there since the 70s, just like he has the best job, just go catch bugs and then write down what you saw. And he has a couple thousand Hymenoptera since 1974, where he wrote down the flower he caught them on. And I went into that data set and extracted a bunch of congeneric sets of plants. So here, each of these is a congeneric set of plants organized by family. Some families have more than one congeneric set. And then on the Y or on the x-axis, you can see that there's big variation in the richness of visitors coming to those congeneric sets. So some pairs have one with quite low richness, some with one with quite high, some have multiple congeners that have different richnesses. These are the frequency of interactions. Now, he wasn't trying to build a network, so all of the network caveats come with, like, these aren't timed, these frequencies aren't necessarily real visitation frequencies, but it seems like there's some variation if you imagine him as like a Brownian guy just catching every wasp he sees, which he isn't. He's focused on specific species. But we want to go out into these flowers and test the prediction that in species from these sets that either have really high visitation frequency or really high historical visitor diversity, that they will tend to be strong competitors, right? If you imagine a meta community where there's new competitors coming all the time in, like the octagon, like, okay, you knock one out, now you got to knock another one out, and then someone else is coming in. There's constantly visitors bringing competitors to a flower. You would imagine that might select for strains that have strong competitive ability. You can imagine another situation where, like, this poor euphorbia that, like, gets visited by one species once, right? If there are microbes there, those microbes probably are pretty good dispersers. And so we'll see if they give me money to do this. I also have some predictions. We're going to build real networks if they pay us to do this and actually measure visitation frequency. So yeah, that's what's on the docket. So do microbes differ in dispersal ability? At what scale? Do pollinators differ? Strains and genera differ. These likely differ in dispersal. So I think the take-home messages here are that deterministic dispersal is definitely important. And it's not just a distance-based kernel. That traits about the patch, and in this case, traits about the flowers, predict dispersal. And that's not surprising, because we know that floral traits shape pollinator behavior. But we've kind of not been thinking about it all that much. That light transmission is this sort of understudied floral functional trait, and it seems like it could shape all sorts of things, including microbes and reproduction, and that there's, like, clear differences in dispersal ability between microbes. So with that, I want to thank, I had to change this slide. I have a lab now, which is really exciting. This used to just be a picture of a bee. All the folks in my lab currently, I didn't tell you about work that Adam Bloom is doing. He's working on plant defenses in a non-native plant. Jonathan Karkash is thinking a lot about sort of context dependence of plant parasite interactions, especially thinking about parasitic plants and their hosts. Fiona is doing all kinds of wonderful work, and she wants to be a microbiologist, and so hopefully we can get her somewhere cool like here. And then everyone who helped with all the other projects this whole time. And I think I have time for questions, probably. Sweet, cool. Thank you all for coming.
So you mentioned sort of propagule pressure, the amount of something as an important factor in dispersal distance. I'm wondering, this is maybe very microbially naive of me, but what else might influence why a microbe has a much greater or shorter dispersal interval?
Yeah, so microbes do all kinds of crazy behavior, right? So there's good history of sort of evidence of microbes being clumpy or not clumpy. So you can do strong selection experiments where you centrifuge microbes and then pour off the supernatant, the clumpy ones stay at the bottom, and then you take those clumpy ones and do it again and again, and they slowly get clumpier. And so all sorts of surface factors that make them stickier to each other, but also possibly stickier, forming biofilms on tongues. This is not my idea. This is largely stuff that folks in Rachel's lab have thought about. I could imagine just architecturally. So the way I drew those yeasts isn't unique. So this yeast actually changes its morphology depending on stress. So in stressful situations, it does this airplane morphology where it becomes these branched clusters. In not stressed situations, it doesn't make branched clusters. And you can imagine those branched clusters might get stuck in the tongue better. So all sorts of physical traits. We just got a wonderful internal grant to get a nanopore sequencer. And so once we've sort of been done phenotyping dispersal and hopefully competition traits in all of these microbes, we might do some long-read sequencing and see if there are correlates there. That's exciting to me, but I'm not my wheelhouse. So, yeah. Yep.
Hi, this is really interesting. I have another maybe microbe naive question.
Yeah.
So it's kind of like a chicken and egg thing. Like, how do you know where the microbes are coming from, or if like a previous visit to a flower, the bee goes back to the hive, does stuff with other bees, whatever.
Yeah.
The microbe comes back out. Like, do you know?
This is like,
I don't know the origins of the
yeah, we should.
microbes?
So like, it seemed like a big problem when I lived here and when I lived in Nevada. I was like, where are the microbes going? And then you go somewhere like Florida and you're like, there's flowers all the time. But I mean, people think they probably live with queens overwintering. I don't know if that it's really well established. It's hard to study queens. They sort of hide away. And do you know, it hurts to have like the world expert here. Like, so I can't be wrong.
We have sampled the queen crops of overwintering queens and they do have a lot of yeast in them. So probably, yeah.
Yeah. In Florida, there's a place for them all the time. So in these tropical, I think one of the things, this difference between yeasts and bacteria in dispersal limitation, I think could be real even outside of the tropical pressure. And I think a lot of what we know is about nectar microbes is centered in California just because of proximity. We know some things from Europe, we know some things from Japan, but I think the tropics are different for sure, and neotropics are different. Just in terms of the number of meta communities, which we know is important for assembly, in terms of the abiotic factors. So I'm kind of excited to be in Florida thinking about this stuff. Yep.
So another, I guess, difference between tropical and temperate regions is, in a lot of tropical flowers, not all of them, but many of them just bloom, the flower is open for one day only and then flower dies.
Yeah.
So I guess these microbial communities have a very short time to develop and they need to disperse quite quickly. Do you see, Amelia, I think it lasts maybe 3 days or 4 or something like that.
Yeah, Rachel actually asked me how long. I don't know how long it lasts. I haven't, this is really true, this like instantaneous open and close, right? And a lot of the plants I've been looking at do that. One of the predictions I have, for this grant, if it gets funded, is I think the actual strongest prediction is floral longevity here, right? That one isn't based on this long-term data set, which NSF wants to fund these sort of like collections use things. So I didn't highlight it too much, but I think floral longevity is really going to be a key factor. And what's interesting about Florida is you can go in the span of a couple 100 miles, like plausible multiple dispersal events, right? For a hummingbird, from subtropics to like things that look like here, like arid scrub, right? And it's really dry a lot of the year. And so I think, I don't know. I imagine that short-lived flowers will have microbes that disperse better, if they have microbes at all. In some short-lived flowers, I know Marshall McMunn, who did a postdoc with Rachel, he did a PhD with Rachel and a postdoc with other folks, he did a ton of stuff here, but looked at Helianthus, and they have florets that open for a short period of time. And they were often very sort of devoid of microbes, if I remember right? I think I remember that, yeah. So I think that floral, it could be that just like longevity is a strong predictor of whether you are a good host at all, right? I don't want to say that like microbes are the be all end all for plant fitness. I think most of the like interactions with pollinators are driven by plant traits. So I made this case because that's what I'm doing, right? For microbes being a big mediator here, but I think that like the sexual function of flowers is mostly shaped by plant traits, including longevity, right? So you would imagine that short-lived flowers need to be much more attractive and would evolve in places where pollination was much more consistent, for example. Yeah.
You mentioned that flowers are particularly stressful environments to live in, and you've done a little bit of research looking at how UV light and temperature might be used by flowers to shape microbial communities. Have you thought at all about how different stressors might kind of interact, rather than thinking about them one by one, but actually the interaction of these different stressors and how that can shape microbial community?
Yeah, the short answer is that like it's hard to do one. You know what I mean? Like we're in this world where like an experiment with one is a lot. But I think you're right in wondering about, for example, like you can imagine that microbes might do something like holding up for the winter, right? When a flower dries down, a microbe might have some phenotypic changes that allow it to persist drying down, right? And you can imagine that those certainly would interact with temperature, certainly would change how they disperse. So I think you're right on. I guess one of the things I try to avoid, because I, like you, like love the minutia, right? Like in this, like interactive effects and abiotic effects, I think are really cool. I think that being careful to step back and say, like, is this important for X, right? Like, is this important for plants? Is there a world in which selection might favor traits that are taking advantage of non-additive effects of floral traits? And thinking that through first and finding the system is the first step I would do if you want to do something like that. Because otherwise, like, everything matters all the time. You can do an experiment, right? Like, you can, and so, because I would do that. I'm like, yeah, that sounds great, let's do it. But I think I would just like, try to pick like a plant that matters agriculturally or something like that. I think blueberries are a good example, which are why I picked. Yeah.
There's a question on the Zoom, or a couple questions from
yeah.
Truman Young. One I think is very similar to the question I asked at the beginning about how microbes are involved in their own dispersal, but the other is whether there are traits of the microbes that affect pollinator fitness. And then a separate question about high sugar content being a form of food preservative and being effectively antimicrobial osmotically. How does that relate to nectar microbes, and why have they not invaded our jams?
Yeah, okay. So let's answer the first one first. Can you repeat it one time?
Yeah. Are there traits of these microbes that affect pollinator fitness?
Yes, absolutely. Yes. And like that's way outside of my expertise, but a ton of work in Rachel's lab has looked at this. So, you know, microbes and pollen provisions are really important. It seems there's like a subset of people who have like called bees like microbivores, right? That like they're getting lots of nutrients from this. I don't know if I would go that far, but they certainly change the chemical composition in ways that bees care about. So these secondary metabolites, for example, can be toxic, especially alkaloids. And so if a flower is, or if a microbe is sort of metabolizing those, that would certainly change bee fitness. The second question was
Sugar content and preservative.
Yeah, so it's what makes these such awesome systems for studying community assembly, because the communities aren't simple, but they're tractable, right? Especially there's lots of microbes in flowers, and I sort of focused on a couple, but the ones that grow to high abundance in nectar can manage that osmotic stress, right? Like, and it is a preservative, and it's why there's not a ton of spoilage bacteria, for example, all the time in flowers. And so, yeah, it's great. It's like they really are meta communities. Like these microbes will grow in sugar water and not reach high abundances on petals. And so it's really, you know, like often we're trying to shoehorn meta community theory into like a continuous landscape, but these are for real meta communities that are limited by dispersal among them. And I think it's a really solid place to test this theory. So,
Sweet.
yeah.
Thank you. That's all. Thanks, Jake.
Thank you.